Hi everyone, my name is Natasha Caminsky and I'm a master's student at the University of Western Ontario in London, Canada. This is my colleague, Eliseos Mucaki, and my supervisor, Dr. Peter Rogan.
We are the authors of a review article recently published in F1000 Research. Our work summarizes all of the literature on information theory-based mRNA splicing mutation analysis.
In this video, Natasha will describe the highlights of our paper.
Our goal is to help you better understand information theory-based analysis, give you an idea of the conclusions that we drew from our review, and help you learn how to apply our methods to your own work. So before we begin, here's the full title of our article. It is part of the Rare Diseases collection of F1000 Research. Alright, let's get started. Our lab is specifically focused on improving ways to understand how sequence changes affect, in particular, mRNA splicing. Splicing is the process of recognizing the exon-intron boundaries and cutting, or splicing, out the introns. Defective splicing can have a number of different consequences, and depending on the mutation context, that can affect gene expression and/or function, and thus result in a deleterious phenotype. Over a decade ago, our lab created an online tool, the Automated Splice Site Analysis Server, based on Shannon information theory. This tool can be used to analyze a mutation associated with virtually any disease or condition. I'm now going to go through our paper in order to get the main points across. This graph represents the distribution of deleterious natural site variants that we came across in our literature review. In total, we found 431 affecting acceptors and 604 affecting donors. As you can see by this figure, the frequency of deleterious mutations reported in the literature is highly correlated with the average information content at that position shown in the top panel by the sequence logos. The next figure is a screenshot of a program we developed called Splicing Mutation Calculator. In this figure, we show that many splicing mutations do not simply result in skipping of an exon, and alternate transcript structures are sometimes the predominant consequence of splicing mutations. In fact, some cryptic splicing mutations do not involve the constitutive splice sites at all. This figure illustrates the ways in which cryptic splicing can be activated. This is the last figure in our paper. We plotted the local distribution of activated cryptic sites relative to the natural sites of the same polarity to discern any trends that might be useful in anticipating where such sites might be concentrated. The bottom panel is a magnification of the region circumscribing the natural site. We actually found that there is no preference for observing activated cryptic sites in either the adjacent intron or exon, and also no preference for activation in a donor versus an acceptor. In fact, activated cryptic sites occurred over a wide range of distances both up and downstream of the natural site. Thank you for taking the time to watch this video. We hope that you find our article useful for your research or diagnostic analyses. If you have any questions, please don't hesitate to contact us at the correspondence address in the paper or the ones that you see on the screen. Remember, no matter which mutation is present in whatever gene, our tools can provide insight into interpreting its impact on splicing.